Hello, everybody. My name is Adam Cankaya. I am a computer science graduate student at Florida Atlantic University in Boca Raton, Florida. And today I'm going to be presenting a project I did for my human genomics class. It's an attempt to use GWAS data from asthmatic patients to predict a lifetime risk probability for other people that express similar variants as found in the GWAS data. So this is a quick summary of the methodology that I used. I started with a disease, a complex disease called asthma. From there, I looked up biomarkers in a medical reference book to get an idea of what chemicals in the body are related to asthma and are expressed in asthmatic patients. From there, I was able to identify genes that code for the chemicals, the biomarkers, and for the other components of the biological pathways that those biomarkers are involved in. Once I had a set of these genes, I was then able to use GWAS data to find genetic variants on these genes that expressed a high significance in asthmatic patients compared to the general population. So genetic variants of these genes that are more commonly found in people who have asthma. For each of these single nucleotide polymorphisms, or SNPs, the GWAS data provides a odds ratio, and using these odds ratios and what we know about the general population prevalence of asthma, we can calculate a lifetime risk probability contribution for each variant, so that if a person expresses this variant, we can say that this variant causes them to have a certain percentage increased risk of developing asthma in their life. So as I said, asthma is a complex disease. This is defined as a disease that has multiple simultaneous genetic and environmental factors going on. It's not just a more simple genetic disease where we can point to a single gene or variant as a cause. It's not a binary disease where we can say you either have asthma or you don't. It can be triggered at certain times. It can be triggered by pollution. It can be triggered by exposure to irritants, allergens. It can be something that people are born with as a child they developed. It can be something that they develop later on. Of course, there are lifestyle factors and confounders, things like obesity and cigarette, tobacco usage, exercise. All these things contribute to the likelihood that someone is going to develop asthma over a lifetime. But it's also genetics. So in the general European population, we know it's about 4 to 8% of the population of adults will have asthma sometime in their life. Twin studies show that the genetic contribution towards asthma is somewhere between 55 and 75%. So it's a majority dominant genetic disease. Yet despite this, what we see from GWAS data when looking, at least when we're looking at the common SNPs, those with a frequency of greater than 1%, and with a high significance value associated with asthma, when we're looking at these sets of SNPs, it can only explain less than 3% of the inheritability of asthma. So the GWAS data, we can only show a 3% the genetic inheritability, yet twin studies show that the heritability should be 55 to 75%. So this disparity is known as the missing heritability problem. It's not just unique to asthma, it's all kinds of diseases that we're looking at GWAS data for. There are some proposed explanations for this, such as potentially the original twin studies are biased somehow, overestimating the inheritance. Another big idea is that while we're looking at the most common SNPs and the most significant SNPs, those are a very small percentage of the total number of SNPs, and that maybe the real bulk of the inheritability are not in these most common and significant SNPs, but are actually in the less common ones, that each one would have a very microscopic amount of contribution to risk, but collectively they would make up that difference between the 3% and the 55%. Of course, epigenetics has to be considered as well. And also, these SNPs that we're looking at are the protein encoding genes. They're not the other areas in the genome that are the majority of the area of the genome, the non-coding regions. So as I said, I started with a medical textbook, Biomarkers in Medicine, Drug Discovery, and Environmental Health. They provide a long list of biomarkers on a disease basis. So I looked up asthma and I collected a variety of biomarkers. These are a list of what I found. These are found in patients, expressed in a higher quantity than in non-asthmatic patients. The interleukins, multiple ones, along with their receptors, cytokines, and filaggrin, as well as the lymphoid type 2 cells. These are have been measured. Their concentrations in asthmatic patients are significantly different than in non-asthmatic patients. So they give us some sort of hint into the underlying biological pathways that compose as the asthmatic phenotype. So once I had this list of the biomarkers, I went on to the OMIM website. That is a great resource. And by putting in chemical names and components of the pathways, you can get back genes, lists of genes associated with that. Some of them are pretty obvious, like the interleukins, which are a type of protein that are released by white blood cells as part of an inflammation response. It's a cytokine. Filaggrin is another protein that's expressed in epithelial cells, which are cells that are on the outer surfaces of the organs, such as inside the lungs. The lymphoid cells are found as part of an immune system response. And then when you're on OMIM, you can also look up directly asthma, and it will give you genes that are associated with it, such as these additional ones. STAT6 is another cytokine, and we have more epithelial cell genes. So right away, knowing nothing about asthma, just looking up the biomarkers and the genes, we can see some ideas of what could be causing asthma. We see inflammation responses, immune system responses, responses of the epithelial cells. These give us hints. So now that we have a set of genes that we believe are associated with asthma, we can follow a methodology that is explored in the textbook I used in class called Exploring Personal Genomics by Dudley and Karzysky. This is a method that, like I said, starts with genes, uses it to find genetic variants in the GWAS catalog, filter them, gather their odds ratios, and then use that to calculate the lifetime risk contribution for each variant. So that if a patient expresses a variant, we can say they have a certain increased risk of developing asthma compared to the general population, or compared to someone who does not express that genetic variant. So to start off with, we go into the GWAS catalog online, we put in our genes one at a time, and we filter the results. We filter by significance, like we talked about the p-value. We only want things that are significantly associated with asthma. We filter by the asthma type. The GWAS catalog is very detailed on their phenotypes. So we have adult asthma, childhood asthma, asthma caused by environmental pollution. Right for now, we're just going with plain asthma. We don't want any kind of any complicating factor in that. And then we sort our results by odds ratio because we want to find the most impactful variants. So we try to find ones that have the highest odds ratio. And then when we're looking at these, we also need to look at the ethnicity. Each variant has different results associated with different ethnicities. Some of them you have to look at the actual studies themselves, the underlying papers behind the GWAS data. Otherwise, in the GWAS catalog, it will provide different odds ratio values for different ethnicities. So we gather about 50 SNPs, 50 different genetic variants, and we further trim those down to a list of 10. And primarily what we do, as I mentioned, we filtered out anything except for adult asthma, no childhood asthma, no sudden onset asthma from pollution. And we also filter out any kind of studies that looked at asthma along with other confounding diseases such as COPD or obesity. But another big thing we filtered out is looking at their linkage disequilibrium. We don't want any genes, we don't want genetic variants that are dependent on each other. We don't want two SNPs that if a person has one, they are very likely to have the other. We don't want to have that confusion in the data. So we look at the linkage disequilibrium and pick a minimum R squared value of 0.8 as a cutoff. We only want things that have a distance of R squared equal to 0.8 or more. I made use of a tool online that you can put into SNPs, and it'll give you a nice chart of the LD for different ethnicities. Again, you need to consider the ethnicities. We decided to go with focus on European ethnicity because that is the primary entries in the GWAS catalog. There's just so many more European entries than anything else, so it gave us the most to choose from. And the idea now is that we have this list of 10 genetic variants. We know their odds ratios, that if a person expresses this variant in their genome, that we know the odds ratio of them having asthma or not. And what we're going to do with that is use that odds ratio and multiply it by the prevalence of asthma known in the general population, the general European population. We're going to assume it's 6%. And based upon that, we can do a pre-test and a post-test risk probability. So you can say before the test, before you know your genetic results, you have a 6% probability of developing asthma over a lifetime. And then you get your test done, you know your variants, and now you have a post-test, which would be the original pre-test times the odds ratios of the genetic variants that you have. So this is the list of the 10 genetic variants that we ended up choosing. As you can see, they're from different genes. They're mostly European, and they're mostly adult asthma. We did end up picking a couple childhood ones just to get some variation in the genes themselves. We didn't want to just have only interleukin genes. We wanted other genes as well. And as you can see, the odds ratios here vary from 1.82 all the way down to 105. And they have different biological roles, but most of them are involved in the immune system inflammation response. So before doing the calculations, I just want to go over a couple assumptions. Really, to do these calculations, you should be using the risk ratio or the likelihood ratio. And I don't want to get too much into the statistics, but they're a slightly modified statistical term compared to the odds ratio. The Dudley textbook that we use goes through the math, show that for odds ratios of around 1.5 or less, that it's okay to assume that they're equal. The only issue is that the odds ratio will overestimate the likelihood or risk ratio by about 25%, but it's a general ballpark. We can assume that they're equal. The reason why we want to make this assumption is that to actually calculate the likelihood ratio or to calculate the risk ratio, you need to go back to the original study and get all of the patient data, the frequencies, the number of people that participated in the study and the controls. It's quite tedious. So by just assuming that the odds ratio is equal to the risk ratio, we can go directly from the odds ratio that's given in the GWAS catalog and directly use that to calculate some probability, some useful probabilities. We're also assuming that the risk is multiplicative, which means that say a genetic variant is for the allele T, and so we're given a odds ratio for the TA heterozygote allele. We would say that TA compared to a homozygote of TT, we would say that the TT has a square the risk ratio. Basically, each alleles either of T is one set of odds ratio. So if the patient has a homozygote variation there and shows two T's, then you're getting two sets of the odds ratios or a square of the odds ratio. And also, we're not considering the frequency of occurrence in the general population. Some of these variants are quite rare. We did aim for about a 1% frequency cutoff, but the GWAS catalog gives data beyond that as well. A person may have one or two of these genetic variants, but the odds that they would have more than that, it starts getting much lower at that point because you're having odds of 1% or less, and you're multiplying them each one. So the probability of expressing multiple rare SNPs simultaneously is not likely for something like asthma that is such a common disease. So, as an example, let's calculate the lifetime probability for a person to get asthma if they express a single SNP from our list. A hypothetical person, an adult, a European, without any known other confounders for asthma. And we assume that they express the single SNP. This is a variant for the filagerin gene that we talked about. It has an odds ratio of 1.27. This is the highest from our list for a European patient. So we know that before the test is done, that a European person would have a 6% chance of developing asthma in their lifetime. And then we have the patient take a genomic test to get back their results for this SNP. We find that they do express it. So now we can say, all right, we start with that 6%, we multiply it by the odds ratio of 1.27, and we end up getting a post-test probability of 8.11%. So this means that a patient who has this SNP has an 8.11% chance of getting asthma in their lifetime compared to a patient who does not have this SNP, which compared to the 6% in the general population, you're looking at a 26% increase of risk for a single SNP. For a little even more extreme example, we look at a second hypothetical person. This time, instead of having one SNP from our list, they have three of them. They have three different ones for three different genes. So we have these three different SNPs and odds ratios of 1.27, 1.25, and 1.24. Similar math as before. We start off with a 6% general population, European population, 6% prevalence of asthma. We do the test, we find out that they express these three SNPs, and now we can multiply that 6% times our three odds ratios, giving us now a post-test probability of 12.56%. So that is more than double the increased chance of getting asthma for expressing these three SNPs. Out of all the millions of SNPs available, just these three could potentially double your risk of getting asthma as if compared to someone identical to you who did not have these SNPs. So, as a conclusion, we started off with a disease, a complex disease like asthma, with multiple factors, genetic and environmental. We used a medical reference book to look up some biomarkers that are found in higher concentrations in patients that have asthma. We looked at, we used some online resources to associate these biomarkers with genes. And then we used the online GWAS catalog to find genetic variants of these genes that are significantly associated with asthma. We filtered them further, filtered them further for ethnicity and to prevent any kind of linkage disequilibrium, so that we were getting independent results for each gene. We knew the pretest asthma prevalence in the general population, and we now have the odds ratios for each SNP. So using that and our assumptions, we can do some simple math to get back a probability of developing asthma. This is an interesting project. There's tons more work that could be done to improve it, such as addressing some of the assumptions we made earlier about, we could do some more manual calculations to calculate the risk ratio and the likelihood ratio, and use those terms instead of the odds ratio to get a more accurate probability. And also the consideration, as I briefly mentioned, about the heterozygote, homozygote. In our examples, we're just assuming that the patient is a heterozygote, a single allele. But in reality, they could be a homozygote as well. We have to consider that. Also, in reality, you can't just look at SNPs in isolation. Obviously, there's dozens of common ones, millions of less common ones. Potentially, some SNPs are protective for asthma. If you don't exhibit a SNP that is a risk, increased risk, that means you exhibit a SNP for a decreased risk. So it depends on how you look at it. Of course, the missing heritability is a huge problem for GWAS. Twin studies show it being up to 75% a genetic disease, asthma, but GWAS studies show it as 3% or less. So there are some other ways we could try to improve this, such as looking at the less common rare SNPs, smaller significance values, SNPs in the non-coding regions. Of course, we also had to assume that our patient is European. We were only really focusing on the SNPs that were found in European populations, but obviously this needs to be improved to address other ethnicities. And of course, we are also right now just looking at asthma in isolation, But even the twin studies show that, a good portion of the risk of developing asthma is not purely genetic, it's lifestyle and other potential diseases. So you really need to address those as well in your final probability calculation if you're going to do something useful for a patient with genetic data. These are the references, the 2 textbooks I used, the OMIM website, and the GWAS catalog. I appreciate you listening, and I hope you learned something. Thank you.